My name is Kaylee Harris. I'm from American Heritage Boca del Rey, and this is my science research experiment, the effect of E. coli K-12 on prostate tumor cells as a cancer therapy. This research took over the place of 48 hours, but it took about 72 hours to prepare the cells and the bacteria and all that stuff, and I'll get into that a little bit later, but first, I'd just like to briefly discuss the purpose hypothesis and a general overview of what the whole experiment was. So the purpose of this experiment was to just create a potentially less harmful cancer therapy and using bacteria. So I applied the E. coli K-12 strand simply because in my prior research, I was going to apply this E. coli strand called Nissl 1917, but it's unavailable in the United States. So I found a literature review connecting the strand I was going to use with this strain, and so I slightly adjusted to use it like this. So basically, I would try to record cell counts after the application of this treatment on these tumor cells to see if the applied bacteria would induce apoptosis, which would label them as an effective therapy. And so my hypothesis was if a treatment of Escherichia coli K-12 is applied to in vitro prostate cancer cells, the cancer cells will experience induced apoptosis. Basically, they will die. And why did I choose this? Well, I chose to do this simply because prostate cancer is common. And in most cancer therapies, there's the use of drugs or radiation, something that is very harmful to the body as a whole. So I'm trying to isolate something directly for cancer, directly to lysis cancer cells. And this could connect a little bit to my future research, which would be the comparison of cancer cell treatment versus treatment on regular cells to see if there is some kind of lack of correlation or correlation between the treatment on cancer versus treatment on human. Because if it works the same on human cells and kills those regular normal cells, then we have a problem. But continuing on, so what did I do? So I had 50 milliliters of media and about 500 microliters of cells. And those took about 5-ish days to culture, which was not very fun. And I split those into 4 T75 flasks of 8 milliliters of media each for about 2 days to let them, reach confluency. And then I had 6 well plates, 6 well plates, with a total of 6 wells. And there's 1.5 milliliters of media and cells in each of the wells. So this experiment actually lasted 48 hours instead of the 3 days, but I observed for that period of time. I did just an additional observation after a weekend passed to see if there was any significant difference. There wasn't actually. Many of the wells dried out. That's a different story. So I had 5 different wells with treatments in them. So there's the 5 microliter plate, 10, 15, 20, 25. And this is all the measurement of bacteria that I put into each of the wells. So originally I had 2 milliliters of bacteria and then used a micropipette and then pipette those specific amounts into each of the wells in each plate. And then analyze them at the beginning, I took pictures and images, and then after 24 hours, and then after 48 hours, and use the analysis with the 24 and 48 hours. So there's some images of what I did. And so they're each labeled on the plates of the amount of bacteria in each well. So that's why the plates are labeled, and then which well I did. And so what I found is that there was a lot of cell debris, especially in the plates with more bacteria in them, which made imaging not very fun in the slightest. And I'll just discuss this in my results section in just a second. But I think the most interesting thing is some of these cell clusters. So I didn't find many cells that were kind of individual. They came in clusters, and then once we got to the 48-hour mark, I found a lot less of these clusters and more individual cells or completely suspended cells, which was great. So my results. was I took the 24 hours and the 48 hours. So these are the average numbers of suspended versus adherent cells in each plate. And I took measurements for 3 measurements for each of the wells in the 6-well plate, and then averaged those, and then made the average of all 6 into these graphs that you see right here. So why adherent versus suspended? Well, in my experiment, we were originally going to record cell counts using hemocytometer, but this did not work out. So I had to adjust to using suspended versus adherent and use imaging to process my data. Later on, I intend on using cell morphology to do further analysis, but at this time, I just don't have those capabilities. So overall, what did I find? What was the whole point here? So after 48 hours, I actually found less cells overall in all plates, but I think this was very significant. Well, not I think, but this was very significant in some of the later plates, the 25 and 20s respectively, because I found a lot less adherent cells. The 25 is a good example, but especially in the 20 plate, I found a lot less adherent cells, and this is very significant because of the fact that the adherent cells are more likely to grow into large-scale tumors, like cell division, all that fun stuff with cancer cells. So that's why that has some form of significance. And the way I would continue with this research would be the treatment of the bacteria I was originally going to use if time permits, and then go into viral studies instead of bacteria, because bacteria can be used, but viral studies are really where the future is going with these kind of cancer therapies, simply because they're more targeted, they're more direct, they're faster, and they're more aggressive. Bacteria, a little slower and much more annoying, and viruses replicate very fast, so they're ideal for the situation. But directly using this experiment, this could apply to further research just using different strains of E. coli, because the K-12 was really the basic strain of E. coli that's not manipulated. It's often used in lab studies. It's the one to go to. But variations, I would examine variations of E. coli with different effects on prostate tumor cells or other more aggressive forms of cancer and more aggressive forms of bacteria. And that's my experiment. Thank you so much.